Hello everyone and welcome to today's live webinar, Where to Go From Here, Next Generation Clinical Mass Spectrometry, presented by Dr. Victoria Zhang, a Vice Chair in the Department of Pathology and Laboratory Medicine at University of Rochester Medical Center. I'm Susie Valdez and I'll be your moderator for this educational webinar. Today's educational webinar is presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning, and made possible by an open educational grant from Agilent Technologies. Agilent Technologies has had no input in the selection of speakers, content, or mode of presentation. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located at the lower left of your presentation window, type that question into the box that appears on the screen, and we'll answer as many questions as we have time for at the end of the presentation. Also, notice that you're viewing the presentation in a slide window. To enlarge that window, just click on the screen icon located at the lower right. If you have any trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window or use that Q&A button and let us know you're having a problem. This presentation is educational and thus offers continuing education credits. Click on the button in the bottom left-hand corner to obtain your credits. Without further ado, I'd now like to introduce today's speaker, Dr. Victoria Zhang. Victoria Zhang is a Vice Chair of the Department of Pathology and Laboratory Medicine at the University of Rochester Medical Center. She is the Medical Director of the Ambulatory Services, Director of Clinical Mass Spectrometry and Toxicology Laboratory, the Associate Director of the Clinical Chemistry Laboratory at Strong Memorial Hospital, and the Associate Professor of Pathology and Laboratory Medicine. Dr. Zhang is an international leading expert in clinical applications of mass spectrometry. Her clinical and research interests are in the clinical mass spectrometry, translational research using omics technology, test utilization, particularly through the laboratory formulary model, and laboratory management and leadership development. She has published over 50 peer-reviewed articles and book chapters, over 70 national and international presentations, and numerous media reports and interviews. Dr. Zhang has been in organizing committees for nearly 20 regional and national conferences with similar number of international and national symposia, workshops, and short courses. She's on several editorial boards in the field of clinical chemistry and laboratory medicine. Dr. Zhang has devoted her efforts into mass spectrometry education for the clinical community in the past few years. She is the founding and current chair of the AACC Mass Spectrometry and Separation Sciences at MS3 Division, the faculty chair of 2 online certificate programs for mass spectrometry, and spearheaded the national mass spectrometry workshops during the AACC and the AACC MS3 annual conferences. She is a faculty chair of the AACC Global Lab Quality Initiative in the Asian Pacific Working Group to lead the efforts to enhance quality and practice of clinical chemistry in this region. Dr. Zhang was also the president of North American Chinese Clinical Chemists Association, the past chair of AACC Upstate New York Local Section. Dr. Zhang completed her clinical chemistry fellowship training at Harvard Medical School after obtaining her PhD in biochemistry and bioinformatics at the University of Minnesota. She is certified as a diplomat of the American Board of Clinical Chemistry. Please join me in welcoming Dr. Victoria Zhang. I will now turn the presentation over to her. Welcome, Doctor.
All right. Well, thank you, Susie, for this very nice and warm introduction. And good morning, everybody, or good afternoon, people on the East Coast. It's a great pleasure to be here to present this and share some, the area that I'm passionate about with the audience. And thanks also for LabRoots to organize and planning for this session. All right, so what we'll talk about today is where to go from here, next generation clinical mass spectrometry. All right, let's see. So the presentation would give, as the title says, more of a 40,000-foot view of what's going on with the current field of clinical mass spec, and then where to go from here. This outline listed 3 items that I will cover throughout this presentation. First is a general overview of LC mass spec applications, so that get everybody on the same page as the main topic and the area of interest. 2 is to discuss the challenges in future implementation of LC mass spec in clinical labs. With that, LC challenges create opportunities, so we'll talk about not future, more of a near future applications of electrochromatography mass spec, which are also coined by the AACC and the MS-Cube division is called next generation clinical mass spectrometry. Is where we're talking about from the current generation to the next generation. All right, this slide shows the clinical mass spec has been a fast-growing field. This is the number of publications that I searched through PubMed actually on September 15th, which is about a month ago, to check since 1991 to current, how many publications has the number of related using clinical mass spectrometry. They were basket in every 5 years. Let's see, I get my pointer here. All right. So this is basically from this is first 5 years and then go up from 1991 to 2016. So if 2016, I can see if this is the projection, we will have about this many publications. So we can really see a very high projection from earlier years, 20 years ago to now. If we really think about even the past 5 years from 2011 to 2015, that's about over 10,000 publications. So average about 2,000 per year. That means it's about 3 to 4 publications per day related to clinical mass spec. This is really a fast-growing field that we have a lot of people dedicated to their career and passion into this field. That also means that we have a lot of exciting developments in this area. So as that being said, I think there's a lot of things that we can and need to think about for the future of clinical mass spec. All right, actually before we go further, I wanted to bring us back a little bit. Although we're talking about mass spec now, it's a very, very hot field. Actually, mass spec itself already has more than 100 years history. This shows a general timeline of the mass spec development. Back into 1899, J.G. Thomas is the first person who created the first mass spec. He also won 1906 Nobel Prize for his innovation. And then we also have different people across the time and devoted their career for the development of mass spec, coming from this to this way, 1950, that's when we talk about quadrupole. This is the area that's got developed. And then also 1984, 85, this area, that's when electrospray ionization and MADI was introduced. And Dr. Zhang and Ms. Tanaka shared the 2002 Nobel Prize for their innovations. So there's a lot, as you see, Nobel Prize involved with the development of mass spec. It has been a great area that attracted a lot of talent. The clinical mass spec actually have not really started until the 1950s. I'll share a little more in the next few slides. So the overall is that we really had a long and rich history of mass spec, and now we're just pushing them to the new area called clinical diagnostics. And from the laboratory perspective or the instrument perspectives, this is what it looked like before. As we can see that this is the original mass spec, and I'll list a few. There's no preference of any vendor or manufacturer. This is just a different models that I search online with the new most popular manufacturer names. Now they're how they look like. We can see it's really night and day. The functions are still there. They also becomes much more powerful and provide much more applications in the clinical side. As so powerful of mass spec, and then we'll see why are we actually using mass spec. This is just to get everybody on the same page as we move forward. You probably already heard this from previous or other presentations through the theory. Just to summarize here, there's a few elements of mass spec that attracts people to bring them to the clinical lab. First one is the high specificity. The high specificity comes from a few factors. One is that the HPLC retention time. Also, we have molecular weight. We also have the multiple reaction monitoring mechanism that enhances the specificity of the identification. For one of the examples is drug conformation, which immunoassay cannot differentiate similar compounds, but mass spec can do that. The second reason is high sensitivity. I also see it's not always the case, it's not universal, but it is true for some analytes, which applied for endocrine testing, for instance, like testosterone. That's utilize this high sensitivity can have a lower detection limit to differentiate, for instance, female versus male, or particularly have more accurate measurement for low testosterone concentrations. And the third part is probably most discussed, which is the quick response to clinical needs. What does that mean? That means that different from immunoassay, which requires a development and selection and perfection of the antibodies, and also through mostly the lengthy FDA approval process, at least for now. We don't know what's going to happen yet for the future. But mass spec provided a much flexible in-house development process, or we call that laboratory development test. With what we need in the lab, we can develop the assay according to a very stringent clinical guidelines, and including state and federal guidelines, and develop the assays in-house to support and provide the needs that we need from the clinical side. Some examples include like the designer drugs, right? Those are changes every day. We're chasing the tails. Those mass spec provided a great opportunity for those. Another example is immunosuppressants. Before we had the immunoassay, those are mass spec played an important role. Another factor is the lower operating cost. I wanted to emphasize the operating side because we all know it's pretty expensive to get a mass spec, but after you get it, actually the operating cost, the day-to-day cost is relatively low as the reagents are inexpensive. So this is the few reasons that we use mass spec for clinical assays. All right, so now the next question is, what assays have we been using for clinical laboratories currently? I just give a few examples here. This is a few very big categories. There's more applications than this, but those are the majority and the major applications right now of mass spec in clinical lab. The title says mass spec is really not just a tool, it actually enhances the clinical practices, and some of them that other technologies cannot provide. But mass spec has provided a unique niche to really help the healthcare and the patient care. The first is the newborn screening. This is one of the early applications of mass spec in clinical lab. Although now we're talking a lot about liquid chromatography, the mass spec here it started with the GC, and there's still many people using that. So as I always say, technology, we use their advantages and limit their weaknesses. And either GC and LC, whichever works the best for you, it is the tool that you can and need to choose. So newborn screening is one of the tools early stage using GC mass spec, and now sometimes it's LC. And then another point is a clinical and forensic toxicology, which involve pain management, drug of abuse, and also including forensic toxicology. It requires different regulations, but the methodologies are very similar. So that's another major area that has been used by mass spec. The third point is a therapeutic drug monitoring. That's also an area that most people are familiar with, for instance, with the immunosuppressant drugs and chemotherapy drugs, or even other antifungal drugs or others. Those are all we can use mass spec to monitor the drug dose and follow up with treatment. Another area that we're all pretty familiar with for mass spec is endocrinology. Vitamin D is one of the examples, testosterone or thyroid hormones that have been used in mass spec. All right, there's also emerging applications. Those include microbiology and I see protein and peptides. I call them emerging after microbiology area now. It's pretty well adapted. It's a fascinating field that to see the adoption of mass spec in microbiology has really changed the 100 years of practice in microbiology overnight by mass spec and created a significant contribution to healthcare by reducing turnaround time. So it's really fascinating. Another area of more credited to chemistry is peptide and proteins. I call them emerging. There's a few applications here and there, and also we have a lot of interest in this area to develop even more. And I have a box called like many other applications, as we see at the earlier slide, with over 2,000 publications every year. Every time we turn around, there's more applications. So it really has been a very, very exciting field. So now as we see, we have current applications, but there's people asking, how do we do more in mass spec? What's really stopping us from doing more? So I wanted to bring this back for people to think about the challenges to bring mass spec assays to clinical practices. It's not universal. Many people want to do it, but not necessarily easy. All right, here are a few things that the gaps that we can identify. One is the high cost for instruments. It typically is capital equipment. It requires a service contract, requires infrastructure, and to get them together, that's a few $100,000, right, for one assay. For one instrument, it's not cheap. Another is the limited choices for personnel. So we need operating staff, we need troubleshooting staff, we need actually people to develop those assays. As they are laboratory developed tests, then we have to do that in-house. So those are personnel that are not readily available. So this is the second limitation. 3 is the complex workflow. It requires a lot of manual process, and it's not automated, and requires special training for the staff and for the laboratory to accommodate that. The third part is regulatory considerations as a laboratory developed test. As we know, there's a lot of discussions from FDA, what's going to happen, how we go about it, and even with the CMS. It is an area that has been heavily debated. We still kind of see where the executive direction, where it's going on. But on the other hand, I think people are also, we know that there's existing regulations on top of that we have to follow. So that's still, an area of kind of unknown. So if I use this diagram to summarize what's going on with the lab, this is basically classic MRI diagram and how to bring mass spec now into the patient side. I'll see we'll have $1, 2 are people. Not only one of them, we need a lot of them. So those are real challenges that are facing us that for the adoption of this fascinating platform. This is what most people, I think, we can pick on those few items. Actually, we also did an interesting survey through the MS-Cube division, or mass spectrometry and separation sciences division, a couple of years ago, to really also assess from our members and the community, how do we generally feel about it? Put some, we're all scientists. We wanted to put some numbers, do a scientific approach to see what are the challenges we have. So we asked the question is, what are the areas that's lack of development here, which means we call them the current status as low. But on the other hand, we also ask what's the question for needs more attention or put a higher priority. And we ask manufacturers, academic representatives, and also regulatory bodies. The result is on the right. It's based on the low current status and plus the high priority. So this is basically the rank we have. Automation comes to first. Automation comes to first. That means the current status is low. And also people think and believe that they should take it a higher priority. So that's why the automation ranked the top. So the same way that we ranked informatics, the education and training is on the third, reimbursement. The fourth, commercial applications. That's really mainly referring to the commercial kits that's available for the lab, and disruptive technologies. And then instrument sensitivity resolution. The third part is the precision medicine. I want to mention precision medicine is one people you feel like that the status is also low, but also there takes a lot more effort to get it off ground. So that's actually, it's a very important area. It just takes longer. That's why it ranks a little bit low on the spot. So I wanted to point out three areas: automation, education, disruptive technology, as those are the things that I will touch more in this presentation. All right, to put them in a different way, this is what I think about. When we talk about now, we have the challenges, and we wanted to talk about the opportunities. Or we see, if we were to talk about the next generation clinical mass spec, what are the areas that we wanted to think about? As you can see, this is actually relatively busy slide. A lot of areas that need attention and can be further developed. So just to go through briefly some of the elements. One is called assay development. From assay matrix, we're moving from qualitative to quantitative assays. Instrument size, we're moving from big to small, which is the miniaturization of mass spec, how we even think about have point of care in a setting so that people can readily available and use that. From analyte abundance perspective, we wanted some for high abundance, low abundance analyte. From the instrument resolution, we wanted them to have a higher resolution and readily available for instruments. And then from the clinical field, we're talking from CP to AP, which is from expanding just from chemistry, actually expanding from even clinical pathology to anatomical pathology. From the molecular size perspective, we're working on from small molecule to large molecules, and the workflow automation. So those are all the pillars that when we talk about the next generation clinical mass spec that need attention and deserves attention for us to study more. All right, here, let's see. And the bottom here is also the training and education for medtech residents, fellow, and pathology becomes a very, very important area, which is I put them at the bottom because they are the foundation of all those areas to even move forward. For the rest of the presentation, I'll just focus on actually those three things and also education, to focus on those areas like what have we have been doing, what is the challenge to move forward, and just give people some, I think, elements to think about where we go from here. All right, from the first one, clinical field, from CP moving to the AP, anatomical pathology side. All this can be seen like tissue imaging. I'll use actually just a couple of slides to talk about a very high level. I see, all basically going to be on the 40,000 foot view of this area, or people call it multi-imaging mass spec. This is essentially the current practice in anatomical pathology. People have the size they get here and read under microscope, either alone or in a group. This is actually the picture of our residents sitting together, discuss what's going to happen. Or I found another interesting picture. I thought that's really cool just to see how people get a consensus to really look at using everybody's expertise to find the right diagnosis for patients. It has been going on for a long time, really provided great values for clinical practice. So the question is that how can we use mass spec to support the development and the applications of in this field? So as we know, this is the area basically is multi-imaging mass spec. The goal or the hope is to replace the microscopes with a multi-top mass spectrometry, as we're showing here. So I'll just, as a very high level to discuss the process, it's actually very, very interesting field. It has a lot of experts working in this area. This slide just to show a general workflow of imaging mass spec, just to see the feasibility of that. And people have been doing that, and we are working on it. I think this is very, very like a feasible area for the future for us to give more thoughts. So on the top, that is the Molotov process, right? We have the ionization process. We have the top to separate ions and then get the spectra at the end. So with a tissue at the front, and we can also sampling at each different spots on each tissue, and also with the tissue slides, we can slice them in a different way. So as a result, we could get thousands or hundreds of the spectra that's showing underneath here. All right, so this is let me get my pointer here so that we know what we're talking about. So this is one of the representative spectra from MODI. We know MODI as when you typically one charge, it does have a bigger molecular weight range to go to here at 20,000. So each spot of that can come with a spectra like this. So one tissue slide can come with like hundreds of thousands of that. And then this can be mapped out of each compound, a molecular weight across 3D dimension to see where they are located on the tissue. So that's how we get the, now we're talking about, let's see here. All right, so we're talking about those slides. So this is the area of the average spectrum of that molecular weight across hundreds of thousands of slides. So put them together. So this, so that we can view the compounds on analyte in this fashion, and then we can identify the boundaries or different differentiated normal versus malignant tissues. So this is the general idea. The samples people have been using either fresh frozen or form fixed parafilm embedded tissues, FFPE tissues. The analytes, with the power of mass spec, we can analyze proteins, peptides, lipids, drugs, metabolites, or even xenobiotics through mass spec. Also, there are, while we're excited with a lot of developments, there are certain challenges of this. Just to summarize at a very, very high level here, one is the detection limit. How low can we go with a molecular weight, or what's the area of the on the tissue, and also ion suppression. When we deal with like whole tissues, there comes with ion suppression. How do we, and other interference. Another is sample integrity. For instance, like a fresh frozen versus FFPE tissues will come with a different level of sample integrity. How do we handle those issues? So those are all very actively studied areas. And also imaging resolution. Is like 2 microns enough, or all of the size of laser, so that we can create enough spectrums for us to analyze, and also the speed of imaging. Although mass spec is pretty fast, but when you're really talking about sampling the whole tissue slide, and also with multiple of that, it takes a lot of time. But also after the imaging resolution, speed of imaging has been increased significantly in the past few years, but there's still room for improvement. Another area, the sample heterogeneity, is just because the cancer, one cancer is different from another. How do we really have enough sample pool to see to develop the patterns for us to use to differentiate the different stages? So that's also a critical component. And by the end of the day, there's another area, which is informatics. How do we handle all this data? All of those are interesting questions, important questions to ask, and also related to image and mass spec. But the field has been developed a lot. It's very exciting just to see that mass spec can go from just chemistry, from chemistry to microbiology, basically to clinical pathology, and then now to anatomical pathology. So this is one area that has been actively studied. All right, so this is image mass spec or tissue imaging. Another area I wanted to discuss is the molecule size. So see, traditionally, mass spec has been used in the clinical side is in small molecules. And now what we wanted to do is move to the large molecules. When we talk about large, mainly it's protein, peptides, or lipids. So this is the area coming from, urine, clinical, or forensic toxicology, as we discussed previously, and now to peptide and proteins. So this area already have a lot of study. I think people know more of this. Actually know, a lot of information in this area. And personally, actually, I started with the proteomics. Now, working into small molecules, now coming back is also have really a special place in my heart in this area. So I wanted to spend, the next few slides to talk about this development and the area and the challenges when we move from small molecule to peptides. All right, so this slide generally talks about the workflow for people also referring that as targeted mass spec in a clear certified laboratory. When we talk about from discussing from small molecules to the peptides, there are typically three components that I would like to separate that. One is the sample preparation to compare what the similarity, what the differences. And then the LC mass spec separation and the mass spec analysis. So in this area, actually, the mass spec analysis is essentially the same. They all use triple quad. And the separation is also similar. That's typically used reverse phase, right, or most commonly used columns. But really, most challenge here between small molecule versus protein peptides is the sample prep. All right, so what does that mean? What does that include? So this slide shows a sample prep workflow. In the clinical lab, what we're used to is working small molecules. This is showing is the small molecule working flow. If you see the slides here, I have two colors here. One is the brownish color. This is the protein and lipids. There's also a bright, more of orange color, and what shooting on your. This is representing small molecules in the patient sample. What we normally do when we work with small molecules is we use a protein crash, meaning get rid of proteins. What we want is what's left on top, which is the small molecule to study. And as we purify that and concentrate that, we put them into mass spec. This is the general workflow. If we're interested in protein, let's see, I move to the next part. All right, great. So we actually wanted to capture those proteins so that we want the large molecules to study, and we wanted to discard those small molecules. So that to achieve that, it requires different tools and technologies. Here listed a few that's typically used for the capturing protein process. One is immunoaffinity enrichment, and we also need protein denaturization and protein digestion and purification. At the end, we also need another final cleanup step. Excuse me. All right. So after the final cleanup step, that's when we send the samples to mass spec for analysis. Or in a different way to view this overall workflow for proteins, it will look like this. Some of the steps could be sooner or later at a different stage, but overall this is just one of the workflows. Individually might be different. So we just use this as an example to show this is pretty, I think, something generic. So one is the protein enrichment. There's so many proteins, different kinds of proteins in, let's see, serum or plasma. So if we were to study one specific protein, we wanted to enrich or extract them. The technology we typically use is affinity chromatography or multi-step liquid chromatography or ion exchange size exclusion, and etc. Those methods we typically use for protein enrichment. And then we have to use denaturization. The natural stage will be difficult for the next step, which is called digestion. So we wanted them to denature and separate the disulfide bonds so that they completely relax in the solution. After that, we wanted to digest the proteins into peptides. In the clinical lab, we don't really measure protein directly. We do have a conversation talking about top-down approach, but generally speaking, so far we measure it on the peptide level. So they're either chemical or enzymatic digestion. One of the most commonly used is the trypsin digestion. And then after that, we will enrich the peptides, and then to separate the peptides from all other, obviously the peptide of interest from other peptides, either use immunoaffinity columns or beads or other technologies. And then after that, at this point, as we know, mass spec is pretty tricky, and most compounds going through this process are not necessarily compatible with the mass spec. We had to do a cleanup, like remove salts, detergents, polymers, solvents, reagents, etc., so that they will be mass spec ready. So it is a very different workflow than what we normally do or we're used to in the lab. So those are a different way to just to list a few things in a different way to talk about the workflow is, the reducing reagents or protein digestion, use a couple examples. So people have to learn this technology to actually apply to the mass spec. Even for people who are familiar with the proteins, majority of them is protein discovery or protein omics. It's not actually still, with a clinical assay, there's still some gap, and people need to narrow that gap to put an assay that's clinical grade. So those are actually different layers of challenges. Here, as we talk about digestion, just to show what does that really mean. So the digestion conditions can have a big impact on assay performance. This is adopted from, reconstructed from paper Dr. Hupniger published on clinical chemistry, talk about thyroglobulin digestion. Here the list are six peptides, all this area, and then based on 3 digestion conditions. They are ranked actually based on the first digestion area condition. Those 3 specifically, 1, 2, 3 on the left listed are not really the discussing point. Just know there are 3 different ways to digest the sample. But what's important here is to see the diversity and variability of the signal from different peptides based on different digestions. For instance, for let's see, the first one. Based on the digestion condition number 1, it has pretty high signal. It's also high in the digestion condition 2. However, the same peptide, when you're really looking at down here, it has a very low signal. Generally speaking, the digestion condition number 3 provided a lower signal. That's not what we want. On the other hand, let's see here. Let's see the 4th peptide. Let me get my pointer. This is not the highest peptide, generally one of the highest signals, but digestion actually number 2 was pretty good. Right, similar condition as this one. There's also lower for among those 6 peptides based on digestion condition 1, but this is a pretty high based on digestion condition 2. So the take-home message here is that different digestion work generates different signals and a lot of variability, and the choice of that will be very important and also determines which peptide we're going to use. This generates a lot of challenges for when we move to the peptide or protein realm from small molecules. This figure actually shows another layer of a complexity of studying proteins. When we study small molecules, that's practically that's one compound, and based on the fragmentation of the compound MRM or SIM to determine the molecule. But when we talk about proteins, there are different areas that we come to consider that's listed on the left side. One is there's more peptide to choose, There's also sample preparation conditions that we need to think about. Also difficult to find the unique peptide to choose from. Also complicated fragment patterns. So here on the right side, if we can see, this is the peptide that's been chosen for thyroglobulin for the measurement of the one peptide for the entire protein. So this peptide is better to be unique and provide enough signals. So this is just think about how many peptides are going to generate through all this, the whole protein. But even finding this one becomes challenging. But even for this one, peptide also have much more complicated fragment pattern as we're showing here. So which fragment peptide we need to use and monitor, this becomes another question. So overall, as this indicated, it's not that easy to identify the unique items for protein assay. So this lists a few things that we talked about before, like also other challenges for protein assay. Low abundance, post-translational modification, we have noctile protein heterogeneity. Those are all other challenges for protein assays. All right, so this is when we move from small to big molecules. Another I wanted to touch base a little bit is the workflow. How do we move from manual into automation? All right, here. So this is the current typical workflow of a manual process, a mass spec assay. Also, so here, actually, LC and mass spec is actually not the limiting step, and I'll show you why. When we have the sample coming in, that's the only pointer I can find. I'll just use this one. It takes about a few hours or even overnight from the sample to get here. And then we through the sample receiving side, or 10 or just say 30 minutes, right? And then go through sample preparation. It's another focus on the time we're talking about. 10 or 30, this is also manual process. And then after the sample is processed, we can go down to the mass spec. LC separation is a few minutes. As we see, we typically put it on the auto sampler and then go directly to mass spec. Mass spec is, we're talking like a milliseconds analysis for each amylite. It's also pretty fast. So we can see I label them into different color. Even so far, we know this is actually the most least manual process in the whole pipeline, right? But after that, we also need more time, like data review takes more time. And then data reporting, if you don't have the interface, that will be more time as well. So although the current workflow, the focus is really for sample prep and before and after LC mass spec, that introduce a lot of LC labor-intensive functions. This is just an example that we have detecting vitamin D, what that means for data review and data reporting. Let's see, estimated we actually have about 300 to 400 samples a day. We report 2, D2 and D3. That's about 600 results. Each one we type in with the 5 digits. We're talking about 3,000 digits per day. And every one of this needs to be checked by another step. So this takes a lot of time. So this one is probably work with Dr. Alan Rockwood, and we just actually published this a couple years ago. So this is the current workflow. So we also propose a couple models just for the fun of that. So if we were talking about automation, what can we imagine? I think this is really not rocket science. It's just something for people to really, it's pretty intuitive. One model we call the standalone model. What that means is like we have the sample receiving side from barcode all packed in one analyzer, and sample prep, handling, liquid handler, and also data review, data reporting. And I put the bidirectional interface separate, as this is actually a very important element to make this more automated than what we have now. It's just interface will save a lot of time. It's more like, if you think about this, no preaching for any vendor. This is one of the Roche analyzers. It's just like a standalone cobus. This is what we use, so that's what I'm familiar with. Another model, I'll call it an integrated model, is what about, we can separate the pre-analytical part as sample receiving, sample preparation, or aliquoting up front, or in one pipeline. And then after that, we put on the different stations. One, the sample, if we need to do chemistry, we do chemistry line. If we need to do hematology, we put on hematology or immunoassays. and maybe mass spec can be one of that. So that would be a really interesting model to think about, and that will certainly help our system quite a bit. And this is also a massive and very impressive pipeline that we can actually find from online. So this is basically the line here. We go to different stations, or we can use the line, let me see, here or this way, or one of them is mass spec. Those are all, I think, interesting ideas to think about. So they're certainly going to solve some of the challenges that we have with automation. I know we have some of the automated, something like this or more of the standalone on the market now. I'm not going to go to the deep conversation on that, but I think we're really making very big strikes in this area. All right, the next few minutes, actually, I wanted to touch base about the training, because this is a very, very important field. Now, we all know it's very challenging. Everybody have trouble getting medical technology to run, particularly for mass spec. So this is really a bottleneck. Beyond everything else, this is a bottleneck for us to moving forward. And when we think about the future of mass spec, this is an area that we have to think about, have to discuss. I also spent quite a bit of time the last few years to really build a community and to share some of the knowledges. All right. Okay, so what are we talking about when we really think about the staff level to run a mass spec lab? So this is I call that ideal structure. We have a couple of people, we call them experts here, sitting on top, and to help you for development of your assay. And then we have another layer of people. They can do troubleshooting. We know that. We need people doing troubleshooting. And we have a great, people that here covering as the operations. This is the ideal. I mean, everybody, I think that I'll be very happy we have this. This, I call that the real life structure. This is, we see the holes here, this, and that. And we're happy if you have even one person can expert at that level to help with the development, but not necessarily true for everybody. And the same is true for troubleshooting. Maybe you have 2 people missing. You want to have 2 more people instead of 1 more. So those are all real challenges in the lab. So the reason being for that is that what we know it's hard to find people, hard to train, hard to even to get people to get to the level that we want. All right, so yeah, here basically very limited training at school for mass spec. All right, but if you are lucky to have somebody, what are kind of the functions that we need to think about at different level? This is what I listed a few things for personnel when they do troubleshooting, right? First of all, they need to be able to follow the protocols for validation. They need to troubleshoot the routine concerns, like, the pressure is up, the pressure is low, right? The column get clogged, all that. They need to know the basic routine concerns. Another is identify concerns with, identify if basically before that's happened, do you have other approaches to identify concern with our different assays? Even be able to, another is separate HPLC issues versus mass spec issues. And also to facilitate training for other staffs and even generally create SOPs and procedures. I all can see, those are the people who need to know as a personnel for troubleshooting, also particularly be able to separate HPLC issues versus mass spec. This is huge. The person needs to have a lot of experience with the instrument and be able to troubleshoot. And also back here, HPLC background is very, very helpful. Majority of the issues are coming from HPLC rather than mass spec. How about experts? The expert level need to know how to do method development and validation, how to bring assays in-house, and they need to know, develop new assays in-house, also training other people in the lab. This is all around basic laboratory develop a test. And also, set up protocols how to have people avoid common issues, and all of that can enter the expert arena. And also, the candidates requires a lot of experience, a lot of attention to detail, and need to be creative. So those are actually very, not really easy to find. I said this before and some people like it, so I'm going to repeat this, is I call it the 10 years experience is not equal to 1 year experience repeated 10 times. What that means is that to become the expert every day, every time we're doing experiment, we have to with the goal to get better. Just like people playing golf, if you just hit the ball every day, right, you won't go anywhere. But if you want to be good, every stroke with the goal to get better. So that is a 10 years experience. Every day you get more, you learn more. We do have a lot of great people actually who are at that level, but also they know, and we know that's not easy to achieve that. This is another challenge for mass spec. With that, actually, even people doing like other assay development for research, they also come in here not necessarily knowing instrument doesn't necessarily mean they know how to develop a clinical assay. There's a whole set of knowledge that people need to know related to the clinical assays, clinical operations, and a clinical grade of assay developed it's different from a research assay. All right, so the question is where to gain those experience. Right? First of all, there's no shortcut for hands-on experience. So I put the hands-on right in the middle, and you can also get this from colleagues, conference vendors, and online trainings. So I wanted to take this opportunity to share a couple of things actually that I've been going to that gain a lot of experience from. One, like the online training vendors have that. We also have the online certificate program, and also the journals. Like say clinical chemistry now, we also have the Journal of Applied Laboratory Medicine, give a lot of information there. Also the conferences and the community, AACC is great. I'm a member of. The division we set up for the goal to actually educate people and help people to know and to learn more of mass spec. And ASMS, that's another area, and also MSACO, as people here, some people know and have made a lot of contribution as well in this area. Those are all the meetings and the conferences I would recommend people to go. Here are actually just a couple of things. Part of that I just wanted to share with people the area that one is that people need to think about what we need to do when we talk about the training. Two is hopefully this is going to be a helpful tool that you can use. We have, like 2 years ago, set up an online certificate program. One is the introductory, another is troubleshooting. For the introductory, we actually started with how even to make a reagent. So people are the training that to do mass spec, we need to actually talk about how you need to make a reagent, how to use a scale, how to running online, make curves, what the HPLC mean, how the mass spec work, how to integrate peaks, and sample extraction, separation, preparation processes, and the dollars and people, all of that we included in the introduction course and with the personnel here on the right corner to show all the experts that shared that experience. We also have the troubleshooting, which cover how to do system suitability, sample prep, HPLC troubleshooting, mass spec, QA, QC, calibration, data review, even lot to lot. Those are more specific things that we or you to think about more training to have. Those are the areas to think about. I'm not saying you have to take this course, or this is the areas for you to think about. And also, with the online learning, this is the area I think is very important. We opened up for people to talk about, go to the labs with the mass spec workshops. With half day we've been doing the last couple of years that I'm proud to part of that and working with the different institutions to even show the same a real mass spec lab, what they do, how they set that up. We got questions like, hey, what's the nitrogen generator, right? Where's the tank? What do you mean you need an emergency power supply? So all of those provide opportunities for people to gain more knowledge. I also wanted to appreciate the organizations that has been very supportive of that. You also use this as illustrating that we do have a very, very generous community to support everybody. If you need a question, you would find people. All right, so with that, I think I use this as a slide to summarize what we've talked about. One is that mass spec has played an important role in clinical diagnostics. I think it will continue to make a very big difference in healthcare. two areas I want to specifically point out: automation and education are big challenges in the further implementation of mass spec in clinical labs. We also have very exciting, it should be next generation clinical mass spec will cover various new applications. Actually, this is the last slide I wanted to leave with everybody when we ask questions, so that to really figure, I'm a very visual person. So this is the things I think about. This is what we have so far. And this is some already there, endocrine, more applications. Microbiology is coming in, more applications going to come out from peptides and proteins. We probably next further along, informatics we're talking about, automation we're talking about. There's already models that are on the market. And then tissue imaging, we talk about precision medicine, miniaturization, and disruptive technology. All of that education training is the bottleneck that we cannot ignore. All right, so I'll end my presentation here while welcome any questions.
Thank you so much, Dr. Zhang, for that informative presentation. And now, audience members, we will start the live Q&A portion of the webinar. If you have any questions you'd like to ask, please do so now. Just click on that green Q&A button at the lower left of the presentation window, type your questions into the box that appear on the screen, and click the send button. We'll answer as many questions as we have time for. So let's take a look at our audience questions that are already coming in. Dr. Zhang, our first question from our audience is, what will happen with anatomic pathology? Will mass spec take over the whole world?
Well, I think that's an excellent question. Yeah, I would like to see mass spec would take over the whole world. I'm passionate about it, but also I think everything has its own place. Certainly, mass spec going to continue to play a very big role in overall. In terms of anatomical pathology, or here particularly the tissue imaging, I think mass spec in the near future will make a huge difference in that area. Will that come next year? I'm not sure. Will that be like 20 years down the road? I think that's probably too far. I think in the near future we'll see huge differences. But on the other hand, this does not mean anatomic pathology won't exist or pathologists won't have anything to do. I think we have so many things that we can do. As a platform, as a tool for mass spec will provide, I think, a lot of value for that, so that people can free it up, do more other things, so that can have the things that can be routine, can be automated, can be processed by the instrument, and then to give that for mass spec to take over, and then our pathology can do other more innovative or interesting other tasks that instrument or technology cannot do.
Wonderful, thank you. Our next question is, you have spearheaded the mass spec division with AACC. Can you tell us a little more about the division and activities that people can benefit?
Yeah. First of all, I wanted to, this has been a team effort, a group effort to get this off ground. There were many people involved. I'm extremely honored to be part of that and working with many people. We've been as a division for 3 years within AACC, and a lot of activities involved with that. I think the overall plan is to set up a platform or a home for clinical mass spec within AACC, so that people coming from different areas like academic hospitals, community hospitals, industry, regulatory bodies, we've been working with FDAs, and government, to have a same platform on the same room, on the same table, discuss the same topics that we all care about, which is the clinical mass spec. And we will have a lot of activities involved with that for our members. Like we have the annual conference. Last couple of years I've been in Chicago, and this year just finished in October in Philadelphia. We're going to continue to have that with basically a day-and-a-half conference, provide cutting edge and also practical information. And like workshop, for instance, I just described a little bit, which provided a lot of value for our members. The online course. I think the third one, I would say you have to basically read, and then see and do the due part, me and a few others have been working on like a few-day on-site training course from SPEC with medical technologists and even residents. We at the University of Rochester is working with our medical technologists training and people with the CDC and Emory. A few very good colleagues are working on that. I know a colleague from Wake Forest are doing on that too. And there's a lot of activities within AACC annual meeting. We've been participating with the FDA regulatory on LDTs. I think really a lot of things going on. I also have so far over 400 members. I hope whoever interested become of that, and any questions, suggestions, we will be happy to listen. And the goal is to help the community and grow bigger and benefit our members.
Wonderful. Thank you, Dr. Zhang. And thank you, our audience members today. We have a really active participation. Can you share more with us about the challenges in training and education? And what do you see the community, what can they be doing and to help as a whole?
Yeah, I mean, as is highlighted here, this is an excellent question. Although I already discussed quite a few of this, I think this is a topic that we need to raise awareness of the whole community. I think the challenge here, in addition to what I mentioned, one is the lack of education in medical technology training program. So we cannot hire somebody out of medical technology or even with bachelor degrees and just to run mass spec. So that's a gap. With the whole community, I think if you know somebody, if you are part of the program, please consider to add mass spec, even like 2 courses, 3 courses, like half a day to your curriculum for people to start familiar with the technology. So I think just a little bit of information goes a long way, so that for people to be more prepared and more ready to do that. 2 is that even the basic technologies, I mean, now the one thing, automation is fantastic. It's a beautiful thing. But it also limited our hands-on training. With the mass spec, there's a lot of training, a lot of hands-on piece. As we see with our online training program, we start teach how to people pipette. With our own training too in-house, we teach people how to pipette, how to measure reagents. So many things are automated. Are reagents ready? Those skills start to lose. I think that's also important for us to keep in mind that we need to keep that going. So, with the whole community, I think we just need to raise the awareness of this technology, of this platform, and provide opportunities. If you are lab doing mass spec, open your doors. Somebody wanted to see, go there, take a look. So I think all of the things community can really help make a difference.
Wonderful. We have time for one more question from our audience, and here's a related question. There are new models of mass spec on the market with the intention to have them automated. Do you think that we will solve the automation problem for mass spec in the near future?
Good. Yeah, very good question. It's very exciting to see, just for a conflict, I'm not going to name the names for the vendor. I think people ask the question, they already know. We see the models, actually launched AACC this year of a automated platform, more like the standalone model that I discussed, which is very exciting to see. I think, we are really moving to the right direction with the automation. But is that perfect? Are we there yet? I don't think so. I think there's still a lot of work to do. Because by the end of the day, how do we prep the samples? How do we have the kit to be ready? How to get people trained to do that? And on top of that is the troubleshooting. We're still talking about HPLC. We're still talking about mass spec. Those are all the things that we need to think about and pay attention. It's not like we put 2 pieces together in a box, then we can call that an automated process. There's a lot more into that. I think that the vendors and everybody have been doing a lot of work. This is a huge, huge step forward in our community to have the model ready for us to evaluate, to look at. I think we're really on the right path. I can see, in the near future that more and more applications with the automated platform in the clinical community, particularly for those that wanted to have that and without the expertise and the staff to support that. Yeah, I'm very optimistic.
Wonderful. Thank you so much, Dr. Zhang, for your presentation. Did you want to provide any final comments for the audience?
Yeah. First of all, thanks to the audience. Those are great questions and the key questions. I just wanted to leave as one note, as this is an area that we are passionate about, and for people actually listening, and it takes a whole village, right? It takes a whole team to move us to the next stage. Clinical mass spec, I call that a big platform. It's not just an instrument. I think it's a platform. We can put a lot of things and add a lot of applications onto that. I'm just very excited to be in this field. I think for people who are thinking about it, please think more about that, because this is an area that's going to grow very fast. And just start to incorporate it in your thoughts, in your plans, how we can use that more effectively in our community and to benefit our society, and also for the betterment of the healthcare. So I'm very excited to continue to work in this field.
Thank you so much, Dr. Zhang. Thank you for your presentation, and also thank you for your important research. I'd also like to thank LabRoots and also Agilent Technologies for making today's educational webcast possible. Before we go, I want to remind everyone that today's webcast will be available for on-demand viewing through April of 2018. You'll receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now, and thanks for joining us. We hope to see you again soon. Goodbye.